All right, let's jump right in. We usually think that for a new drug to get approved, it's got to be this massive, revolutionary breakthrough, right? A total game changer. But what if I told you the path to approval isn't always about being better, but about proving you're just good enough? Today, we're going to get into the really surprising science behind how that actually works. And this whole thing kicks off with a really tough ethical question. Picture this. You've got a new drug for a serious heart condition. Normally, you'd test it against a placebo, a sugar pill, to see if it works. But there's already a treatment out there that saves lives. So, can you ethically give a group of sick people a useless pill, knowing that the existing drug could help them? The answer is a hard no. So, what do you do now?
Well, this exact situation is what scientists and regulators call the placebo problem. It's when that gold standard for testing a drug, the method everyone trusts, is suddenly off the table.
Yeah, the classic setup, it's so beautifully simple. It's called a superiority trial. Your only goal is to prove, with solid numbers, that your new drug is better than nothing. If the new drug's effect is clearly greater than the placebo's, boom, it works. It's clean, it's direct, it's powerful.
But here's the massive roadblock, and this is a direct quote from FDA guidance. When an effective treatment is already out there preventing, death or serious permanent harm, you just can't use a placebo. It's unethical. You can't withhold a potentially life-saving drug from people just to run a clean experiment. And this forces researchers to get creative and find a whole other way to prove a drug is effective.
And that other way is a really clever solution. Instead of trying to prove that your new drug is better than nothing, you switch your goal. Now you just have to prove that it's not unacceptably worse than the current champion drug. And that right there is the entire world of the non-inferiority trial. So you can see the two paths right here, side by side. On the left, the superiority trial. That's just a straight-up race against a placebo. Simple. But look at the right. The non-inferiority trial is totally different. The new drug isn't racing against a placebo. It's being compared to the existing effective drug, the champ. And the goal isn't to win the race. It's just to prove that the difference between them is smaller than some predefined amount we call M, the margin. So the whole point, the big takeaway, is that a non-inferiority trial is specifically designed to show that a new treatment is not materially worse than the one we already have and know works. It's all about preserving a benefit. Okay, but how in the world do you define not materially worse with numbers? I mean, that sounds kind of vague, right? Well, it all comes down to drawing a very, very specific line in the sand. And that line is called the non-inferiority margin, or just M. And honestly, this is the single most important number in this whole process. It is the absolute maximum drop in performance the new drug is allowed to have compared to the old one and still get a passing grade. It is the official, pre-agreed upon definition of good enough. Now, setting this margin isn't just guesswork. It's a super rigorous process. First, scientists dig through all the old studies of the champion drug to figure out its entire historical benefit over a placebo. That entire effect, that becomes something called margin 1, or M1. Now, if your new drug is better than losing M1, you've at least proven it has some effect. It's better than nothing. But that's not good enough. So, and this is the crucial step, experts use clinical judgment to set a much, much stricter line, M2. This is the largest loss of effect that doctors and patients would actually find acceptable. And this is what success looks like. The new drug's performance, including that fuzzy area of statistical error, stays completely on the safe side of that M2 line. It has successfully shown it's not unacceptably worse. Non-inferiority is demonstrated. Check. But if that range of possible outcomes even touches or crosses the M2 line, the trial fails. Now, this doesn't automatically mean the new drug is worse. It just means we can't be sure that it isn't. The uncertainty is too high, and the potential risk to patients is just too great. Non-inferiority has not been shown. Okay, but hold on. There is a huge, massive assumption baked into all of this. For any of this to be meaningful, we have to be sure that the champion drug, the one we're comparing it against, was actually, performing like a champion in this new trial. This brings us to a tricky concept called assay sensitivity. Think of it like this. Was our measuring stick in this experiment even sensitive enough to tell the difference between a great drug and a bad one? That's assay sensitivity. It's the trial's basic ability to distinguish something that works from something that doesn't. Which leads to this kind of mind-bending question. If you don't have a placebo group to compare against, How do you really know the champion drug didn't just have an off day? I mean, what if, for some weird reason in this specific trial, neither of the drugs worked? If you showed they were similar, what would that even prove? You'd just be proving that 0 is not much worse than 0. It would be totally meaningless.
So, to get around this, regulators rely on something called the constancy assumption. They build trust in the result by making sure 3 things are true. 1, the historical evidence for the champion drug has to be absolutely rock solid. 2, the new trial has to be designed to be almost identical to those historical trials. Same kind of patients, same dose, same goals. And 3, the trial itself has to be run with incredible quality, because any sloppiness could accidentally make 2 different drugs look the same. So we've gone through all this complex science and these tricky statistics, but why does any of it matter? What's the actual real-world payoff of proving a drug is good enough instead of better?
Well, this is where it becomes incredibly important for actual patients. A non-inferior drug might be just as good at fighting the disease, but have way fewer side effects. Or maybe, and this is huge, it's a single pill you take once a day instead of a painful injection. It could be a safer option for certain groups, like pregnant women, or it could be a lifesaver for someone who develops a resistance to the first drug. Good enough on the main goal can be a massive win for someone's quality of life. So, at the end of the day, a successful non-inferiority trial really accomplishes 2 critical things. First, it rigorously proves the new drug has a real effect. It's definitely better than nothing. And second, it shows that it holds on to a clinically meaningful chunk of the benefit we get from the current standard of care. And that really leaves us with this final thought. In medicine, maybe the goal isn't always to be objectively better on a single spreadsheet number. Maybe the goal is to provide a better choice for an individual person. And it really makes you think, in a world that's full of complex trade-offs, how often is good enough actually the best new option we have?